Morning. Welcome to week 2 of Science on Saturday. For those of you who were here last week, welcome back. And for those of you who are joining us for the first time, welcome and hope that you will be with us throughout the entire series. My name's Andrew Zwicker, and I'm the head of strategic partnerships and public engagement here at the Plasma Physics Laboratory, and have had the very, very deep honor of being the host for, I think, about a decade now. I'm not quite sure. We've named, as many of you know, the lecture series after a longtime host, Ron Hatcher, who passed away tragically a while ago, but he had done it for about 16 years and been the longest host. We've lost track. We believe this is the 40th or 41st year, we're not quite sure, in a row of bringing cutting edge science to science-curious people of all ages. So we are just so happy that you're here. We missed you. We always miss you. So for those of you who are here, we ask a couple different things, and I'll just go through some of the health, safety, and logistics. Masks are always optional. Hopefully we're coming on our way out of sort of the little bit of a COVID surge that we saw. Isn't it amazing what today's topic is as we talk about viruses? If it ever changes, and we here at the Plasma Physics Laboratory were to go to mandatory masks, the lectures would continue, and we just would ask everyone to mask up. But now we're in a mask optional situation, whatever makes you comfortable. Questions we're going to do at the end. If you're here in the audience, we ask that you wait until a microphone is handed out, and then we'll try to get to you. And for those of you on Zoom, if you use the raise hand function, we will do our best to try to get to some of you as well. We have speakers from all over, not just the country, but all over the world listening this morning. This is why I know you're all here, is not for Professor Adams, but is for the, but it's good to pretend like you love science. So it's my pleasure to introduce Professor Sandra Adams, who has a bachelor's degree in biological sciences from University of Georgia, where she taught 3 years in public school. She got a master's of education in secondary biology education from Georgia State, a PhD in science education from the University of Pittsburgh. That's where my daughter went. She has served on the education faculty at Birmingham Southern College, State University of West Georgia for several years. She enrolled at Georgia State University, completed a second PhD in molecular genetics. When are you getting your third PhD? And I guess we'll go through the 3 questions. Would be, when do you sleep? And her doctoral research focused on RNA recombination in rubella virus. She's been on the faculty at Montclair State University for 22 years, holds the rank of professor in the Department of Biology. We were just talking about this, just recently retired after 22 years, so congratulations. She has a research laboratory with various natural products. I can't say these words. Analyzed for antiviral properties against herpes simplex virus 1 and 2, Synbis virus, and enterovirus 69. The natural products tested include extracts of polyphenols, you can tell I'm a physicist and not a biologist, from green tea, black tea, embellin, curcumin, and other curcuminoid compounds. Yes. I cannot wait to hear your lecture so I can do better than this. She's mentored numerous undergraduate and graduate students in research. Her teaching responsibilities include introductory biology courses, virology, immunology, and biology pedagogy courses. Would you please welcome Professor Sandra Adams?
Thank you.
Thank you so much for doing this and allowing me to butcher a couple of biological terms. So first question that we always ask is, would you share with everyone, when did you first get interested in science?
I think I've always been interested in science, and I was thinking back all through elementary, middle school, well, it was junior high then, and high school. Science was one of my favorite subjects always, and all of the sciences, biology, chemistry. I even loved physics. So, even loved physics, yes. Yes.
Okay, so you've loved science always.
Yes.
But at some point from when you were in elementary school up through one of your PhDs, I'm sure there were many teachers who were influential, but would you share with everyone one in particular?
There was one teacher in particular, well, I had 2, because one, when I was in high school, I took every math course and every science course that was possible. So there was a math teacher, Mr. Jenkins, who uh, made it fun, made it challenging. And then my chemistry professor, uh, Mr. Elmore, and he's now Dr. Elmore, same thing, made it challenging, but made it fun. But what they both did was make me know that this was something that I could do. And then when I entered the University of Georgia, my first biology course was cell and molecular biology. And I loved it. And I knew for sure that that was the area that I was going to go into.
That's wonderful. Okay,
Mhm.
so when you're not doing science and teaching and mentoring,
Mhm.
something, a hobby, a passion, but not scientific.
Okay. I won't call it a hobby, I'll call it a passion. I love the theater. And as often as I can, I will go into the city, see a play, see a musical. That is truly enjoyable for me.
Well, don't give away your shot as
Yes. I know that
a
line.
few people got that, not everybody.
Yes.
So would you please welcome Professor Sandra Adams, plant extracts and their potentials antivirals.
Thank you. So we need to start today by making sure everybody has a good understanding of what viruses are. And so many of you know, you have an idea in your mind about what viruses are and what viruses do. So let's look at some characteristics of viruses. Viruses are everywhere, everywhere. All living organisms contact billions of viruses every day, billions of viral particles every day. And one characteristic that you know about viruses is, of course, that they cause disease. Viruses definitely do cause disease. And another problem that we have is that viruses can cross species boundaries. That means there can be a virus that infects a bat, that can then infect another mammalian host, that can then infect humans. So viruses can cross species boundaries. But there's some good aspects as well, because they can be used as tools to study biology, to learn more about molecular biology. At Montclair State, we have a program that's partially funded by the Howard Hughes Medical Institute, where students isolate bacteriophage. And bacteriophage are ideal to learn more about molecular biology and recombinant DNA technology. So what are viruses? Viruses are parasites. They are what we call obligate intracellular parasites. This simply means that they rely on their host for replication. They cannot survive without a living host. So all aspects of their reproduction must take place within living cells. And when we look at viruses, viruses are everywhere, and there are billions of viruses. There are viruses that infect mammals, there are viruses that infect humans, there are viruses that infect plants, every living thing. And I put this slide so that you could just see the variety of genomes, or what the DNA and RNA of the virus look like, or what they're composed of, but also the fact that they're very specific for their host. So we have billions of viruses, but they're very specific for their host. Plant viruses, animal viruses, any fungi. So everything can be infected by a virus. And I also wanted you to see they differ in terms of appearances. So not only is the genetic material different, the outer structure is different as well. At the beginning, we've got a bacteriophage, and you've seen that. That's what an image that you see quite often. Then there's a plant virus in the second panel. That's the tobacco mosaic virus. Then a rhabdovesicular stomatitis virus. That's in the same family as the rabies virus. And then the human rotavirus. And you see the terms enveloped and non-enveloped. That means some of them have a membrane around them in which proteins can be embedded. Some are what we call naked viruses, just proteins on the surface. So how do we battle viruses? How do you battle virus infections? So there are two routes that we take, either developing a vaccine or an antiviral. And not all viruses have vaccines. You know that it takes a while to develop a vaccine, even though we now have a really good technology to get them out much faster. So let's look at what vaccines do. There are quite a few misconceptions about what vaccines do. Vaccines cannot prevent a virus from coming in. But what vaccines do is they trigger an immune response. And once a vaccine enters a cell, and that cell has seen that vaccine for the first time, it takes a while to build up a good immune response. There are cells that target the virus initially to try to keep the numbers low until that viral pathogen can be identified and a message then sent to what's called the adaptive immune system to trigger the production of antibodies and cells that can kill the virus. So that takes a long time to develop, a long time meaning several days. But while that's about 7 to 11 days for you to see that begin. And what's the virus doing at that time? The virus is replicating. So while that virus is replicating, you have an immediate response from other cells, again, to target, to identify the virus, but also induce inflammation, fever, other symptoms to try to bring that under control. And so following, again, that initial encounter with a virus, the goal is to identify that virus early so that the immune system can then, once it sees that pathogen again, react much more rapidly and with a stronger and more robust response. That's the goal of vaccination. And out of all of the human viral infections, you're probably going to be surprised to find out, but there's been only one human disease that's been eradicated, completely eradicated, and that was smallpox. And that took place because a massive vaccination campaign and the fact that humans were the only host. So that's a prerequisite for eradicating human viruses, one host and a vaccine. So when we, again, candidates for eradication, two features, single host and a vaccine. And that vaccine must induce long-lasting, lifelong immunity. Now, antivirals are different. Antivirals will target some process that's essential for the virus to replicate, either something on the surface of the virus or something inside the virus, such as a viral protein, a polymerase, which is the copy machine of the virus. So something that would target either an enzyme that's necessary for replication or something that's on the surface of the virus. You've seen images sure you've seen images of the SARS coronavirus too, with the spikes on the surface. And so they target something on either the surface of the virus or inside. So once that pathogen is identified, once you get the sequence, then vaccines are possible, and we can find out more about or look at ways to develop antivirals. But there's also another caution about antivirals. The antiviral must target the virus and not hurt the host. So in my lab, this is what we're looking at. We have two things that we have to focus on initially. One is, will this compound that we're looking at hurt the host cells? And if so, we can't use that. Second is, can it effectively stop some process in the virus from replicating? So in my lab, we looked at natural products, plant extracts. And so I started, it was mentioned in the intro, in my doctoral program, we worked with rubella virus, and there's a vaccine for rubella virus. And so rubella is a good target for eradication. The limitation, again, is if you can get everybody to get vaccinated and reduce the spread. And I brought in a different virus because not rubella, once I got to Montclair State, it was called Synbis virus. And Synbis virus has RNA for its genetic material. When we look at the genetic material, you either have DNA, which has double strand, very stable, or RNA, which can also hold information. So I looked at another RNA virus. It has an envelope, which again is that membrane that surrounds it, in which glycoproteins are embedded in the membrane. And I was working with this in the lab, and I had an undergraduate student who wanted to do research with me. And I said, well, I have this Synbis virus. And he said, is this the virus that causes Synbis fever? I said, yes, it is. And he said his grandmother used to give him turmeric to treat Synbis fever. And I said, okay, let's see what the active ingredient in turmeric is. And that's curcumin. And so that was one of the first plant extracts that we started working with in the lab. And so when we look at natural products, we're looking at extracts from plants. And I'll show you a diagram of a polyphenol. Poly meaning many, phenol is a type of ring. So many of these extracts are polyphenols. And curcumin is one from a plant extract. And then we also worked with various tea extracts. And someone else brought another plant product to me called embolin. So we did some work on embolin. And my last master's student had an idea about garlic extracts. So there is a compound in garlic called allicin, and black garlic is a little bit different because it's much more fermented. So her question was, is black garlic antiviral? So what we do, again, it's a teaching process. Let's see. So we investigated that. And then one of the other last compounds, I call those curcuminoid compounds, hispalons. So that my final project was with hispalons. So when we look at the diversity of viruses, when you look at the genetic material, some viruses are DNA viruses, some viruses are RNA viruses. So in my lab, I worked with two DNA viruses, herpes simplex virus 1 and herpes simplex virus 2. We also worked with two RNA viruses, Synbis, which I just mentioned, and enterovirus 69. That one's completely different. Well, they're both RNA viruses, but EV69 is what we call a naked virus. It's in the same general family that the polio viruses are in. And then Synbis virus, another RNA virus. So I spent most of our work was targeting HSV1 and HSV2. And I've given you the characteristics of both. Herpes simplex virus 1, transmission mainly oral to oral. So oral meaning from the mouth and so and lips. And then, but if you look at the problem, 67% of the people worldwide have come in contact with HSV1. And if you think about it, one problem with that is it never goes away. It develops a latent infection. So you're infected initially, you have an outbreak in what are called epithelial cells, but then it's going to go dormant and it hides for a while in nerve cells and it can be reactivated. HSV2, primarily sexually transmitted, 13% of people ages 15 through 49, 491 million people worldwide have HSV2 infection. So it manifests itself as genital lesions, sores in the genital area. Again, lifelong infection. So with lifelong infection, the problem is they're not quite sure what triggers the resurgence. But when it does, those lesions are painful and it's shedding virus. And this is what we look at. When you have those outbreaks, you're releasing virus that can then be transmitted to other susceptible hosts. Now, the RNA viruses that we worked with, again, Synbis is an envelope, and by envelope mean have a membrane, and it's vectored by mosquitoes. Two species, particularly Culex and Culiceta mosquitoes, and then examples of the diseases that are spread, but usually fever, rash, joint symptoms, particularly wrists, hips, knees, and ankles. EV69 is considered an emerging virus, first isolated in the US in 1959. Little is known about the pathogenesis. In other words, what happens once an infection. So there's still a lot of research that needs to be done on EV69. And I'll be honest, this was the most difficult one for us to work with in the laboratory because when you work with a virus in the laboratory, you have to find a cell line that the virus can grow in and replicate efficiently and generate a large number or high titers of virus. And so EV69, there are some cells that it grows in, but does not grow in as many cell lines or as easily as Synbis and HSV. So when we looked at HSV in particular, there are antivirals that are on the market to treat herpes infections. The most common one is acyclovir. And I'm showing you the structures, and you see those rings. And so acyclovir and derivatives of acyclovir or analogs of acyclovir are used, and they're very effective in treating HSV infections. But there's a problem with that as well, and that you will find with just about with many of the antivirals. After a period of time, the virus develops resistance to the treatment. So even though acyclovir is an effective treatment, it's not 100%. So we're always looking at, and we know how acyclovir works. Acyclovir targets the replication machinery, the polymerase, the copy machine of the virus. So that's one mechanism. So if we're going to look at antivirals that might work in other modes of action. So again, acyclovir and its analogs target the replication machinery, specifically the polymerase. Now I'll begin with, I'm going to tell you about the work that we've done in my lab, and the majority of the work has been done by students. I consider myself a teacher first. So I'm a researcher, but I'm also a teacher. So in my lab, my goal is to teach students, prepare them to also work with viruses and to understand what they're doing, the mechanism. So curcumin was the first one that we mentioned, and it's extracted from a plant, Curcuma longa. And you'll be interested in knowing this plant is a member of the ginger family. And you're familiar with a product in which curcumin is the primary compound. And again, that's turmeric, the spice turmeric. So curcumin is an extract from the spice turmeric. And other research has shown that curcumin is antioxidant. It has anti-inflammatory and anti-tumor properties. There are limitations with curcumin as well. One is that it is insoluble in water, and it is very soluble in polar solvents, such as ethanol and DMSO, which is dimethyl sulfoxide. So many of our extracts, we either suspend in DMSO or ethanol. So again, the limitation with curcumin is that it has limited bioavailability. But I want you to see what we found about curcumin. And as I said at the beginning, you want to make sure, first of all, that the compound will not hurt the cells. And so we start out, so this is when I knew I was going to do that. Okay. This is a 6-well plate. So we will plate the cells in a 6-well plate. And when we plate the cells in a 6-well plate, then that view is large enough for us to look under the microscope. So we will use increasing concentrations of the compound initially in a 6-well plate, analyze it by looking at it with an inverted microscope where the lenses are at the bottom. And so you're looking at the bottom of the dish at the cells. And then we used curcumin with various concentrations ranging from 10 to 100 micromolar concentrations. And then after we looked at it under the microscope to see if there was any damage to the cells, we follow that up by quantifying the number of cells that survive. This is a 96-well plate. And so we could use various concentrations in each row. And we did what's called a cell proliferation assay, whether or not the cell can continue to replicate. Now, this data is convincing. So you always use your controls, and the controls would just be pure media. Of course, the media is not going to hurt the cells, so we have 100% viability with the media. We also want to make sure, since the curcumin was suspended in DMSO, that the DMSO at that concentration was not toxic to the cells. So we can see that. But look at what happens as we increase the concentration of the curcumin. Percent viability on the y-axis, concentration on the x-axis. What do you see? You see, as we're increasing the concentration, viability is being reduced. So we were able to identify the right concentration that we could use to determine if it was antiviral without hurting the cells. And so then in the 96-well plates, we treated the virus with 30 to 50 micromolar concentrations, treating the virus. And this was effective with both HSV1 and HSV2. And these were the results. And we wanted to know how successful it was at inhibiting the virus from replicating. So we looked at percent inhibition. So our highest percent inhibition came when we treated the virus with 30 micromolar concentrations of curcumin, HSV1 in the blue, HSV2 in the green. And so with increasing concentrations, again, the lower percent of viability. So our conclusion was that curcumin at 30 micromolars can inhibit both HSV1 and HSV2, worked more efficiently with HSV1. And how do we quantify it? And I wanted you to see this. And so I've talked about counting viral particles, and we're looking at billions of viral particles. So I trained students in the laboratory to do what's called a plaque assay. In that 6-well plate, we did what's called 10-fold serial dilution. You start out with 100% of the curcumin, and then, well, 100% of the virus, and then we did 10-fold serial dilution. So then we would take 10, say if it was in 100 microliters, take 10 microliters, put it in 90 microliters of PBS, a solvent. And so then we would continue to reduce the concentration. And then we wanted to do that until we got to a point that we had countable plaques. Because if it had 100%, it would have lysed all of the cells. And where you see purple, those are living cells. Where you see, those are the living cells stained with crystal violet. But where you see these areas of clearing, that's where the virus went into the cell. We overlaid with agar so that it could not hop to another cell, stabilized it after a certain period of time, stained the cells with crystal violet, and then we could count the number of plaques. That gave us what's called the titer, T-I-T-E-R, the number of plaque-forming units per milliliter. So that was initially how we quantified the number of viral particles. There are also other ways that we can do that using a reagent in a 96-well plate. So once again, where you see the clearing, the areas of clearing, that's where the virus has gone into the cell, killed the cell. And that's what happens. When a virus infects the cell, that cell is gone. There is no saving that cell. The cell is gone. The virus goes in, essentially takes over the machinery of the cells. It is going to use the replication enzymes that the cell would have used to make more copies of its DNA. The virus has hijacked that. So the virus is now using that cell to make more copies of its genetic material, and it's sending a message to make more viral proteins. So it copies the genetic material. It has the cell make viral proteins. It has included the instructions for assembling the virus. So the virus will assemble in the cell, then it bursts through the cell and lyses it and releases other viral particles. And that's what you see there, where the virus has lysed the cell, killed the cell, with the goal of releasing it to move to other neighboring cells. And so we see what curcumin did in reducing the titer, the number of virus particles. And so the untreated titer was 2.4 times 10 to the 8th particles per milliliter, or plaques per milliliter, plaque forming units per milliliter. When we treated the virus with 30 micromolars of curcumin, we reduced the titer to 10 to the 5th for HSV1 and HSV2 as well. So we had a reduction in titer. And then we looked at the percent of inhibition. We also wanted to find out not just whether the virus can, whether the compound can reduce or stop the virus from replicating. We need to figure out when it did that. So we looked at, did it occur during attachment? Did it occur during penetration? Did it occur after the virus got into the cell? So we wanted to look at the life cycle of the virus. And so what we found was, in terms of absorption or attachment, once you put the virus in and it contacts the cell, did curcumin block the ability of the virus to get into the cell? That we found to be true. So it blocked absorption. But with curcumin, it was not effective at blocking penetration. So our conclusion was that at the very early stage, when you put the virus in contact, well, the treated virus in contact with the cells, it could block it from getting into the, attaching to the cell. So 30 micromolars was more efficient, and then 30 micromolars could inhibit both HSV1 and HSV2 in cultured cells. And the vero cells, I didn't tell you what vero cells were. We work in the lab with 2 different cell types. Vero cells are monkey kidney cells. I also used A549, which were human lung fibroblasts. So this study was carried out in cultured vero cells. So the curcumin at 30 micromolars, not cytotoxic. It did not hurt the cell, but it could block the absorption. And then Embolin. Embolin was brought to me by a chemist at Vassar who was interested in whether or not Embolin would work well against HSV. So I assigned a student this project. And so when we look at Embolin, it's derived from berries of the Ambelia ribes plant that's found throughout India. Embolin, its structure is shown, and then you see an image of the plant. Anti-inflammatory, antioxidant, anticonvulsant, antidepressant, antihelmintic, those are worms, antimicrobial, targeting bacteria, anti-cancer, blocks the proliferation of cancer cells, and previous research in another lab showed that it was effective against Hep C and influenza A when applied directly to the virus. So using this information, we looked at whether or not we could treat HSV with Embolin, or whether or not we simply, and whether or not we can apply it to cells. Same protocol. We have a standard protocol using the 6-well plates to look at cytotoxicity. We wanted to make sure the Embolin was not cytotoxic to the cell. So that's always the first thing that we did. And so based on previous literature, we started with 10 to 70 micromolars of the Embolin. And then in the 96-well plates, we looked at cell proliferation as well as microscopy. But we also needed to check to make sure the DMSO, even though we saw previously that DMSO was not cytotoxic, 2% DMSO was not cytotoxic to vero cells. So when you look at the results, we used what's called an MTS assay, which would quantify proliferation, again, the ability of the virus to replicate after Embolin was placed on the cells for 1 hour. We would leave it on the cells for 1 hour and then look at the effect. And so those are the results. And you see the bar graph again with the percentage of proliferation and the increasing concentration. So Embolin, we concluded, was not cytotoxic to cells at concentrations up to 70 micromolars. Now, I've mentioned microscopy. So I want you to see what my students can see under the microscope. This is just with a normal light microscope. And so when you look at this is just a monolayer of vero cells that you could see under the microscope in nothing but the media. So this is what the monolayer of healthy cells looks like. Now let's infect that cell with virus. You see the difference. The monolayer is gone. The cells have rounded and lifted. And if you could see it moving, they'd be floating on the surface because the virus, again, kills cells. When it kills cells, that monolayer is gone, they lift up and they're floating. So you see round floating cells. So then we treated them with 50 micromolars of Embolin. Compare this monolayer to the monolayer in media. Healthy monolayer. And then, of course, we wanted to make sure that it's not the DMSO. So 2% DMSO also not harmful to viral cells. And this is one of my favorite assays. The virus that we work with in the lab, it's modified to include the sequence for expressing the green fluorescent protein. So once the virus gets in, so the process again, the virus is going to get into the cells. It's going to use the cell machinery to make more copies of the DNA, but it also makes more copies of the virus protein. So when we put in that I didn't put it in. So when we use the virus with the GFP insert, the green fluorescent protein insert, protein synthesis occurs later in the process. And this is called a DAPI stain, and that's staining the nucleus. So that with that blue stain, you can see the nucleus of the viro cells. Look at where you find the green in the cytoplasm. If you've, the protein synthesis machinery for cells is in the cytoplasm. They're called ribosomes. So the message is delivered to the ribosomes to make these structural proteins of the virus. As it's making the structural proteins of the virus, it also makes the green fluorescent protein. So it's really cool to look under a fluorescent microscope where you can see the DAPI stain in blue staining the nucleus, and then you also see the green being expressed from the green fluorescent protein. So now, after seeing that, let's compare it to what happens when you treat those cells with embolin or treat the virus with embolin. Look at that. When you see the nucleus, but not the GFP, not the GFP. And the next step again, how does it work? How does it work? As I said, we're going to look at the replication cycle. We're going to look at attachment, penetration, attachment when the virus will bind to the surface of the cell, to a receptor on the cell, penetration once it gets into the cell after a certain period of time. Then we treated it with the embolin, and then post-penetration after a certain period, longer period of time, can it stop or inhibit replication then? So we looked at various time periods. So when we treated HSV1 with the embolin, the results were that it inhibited binding. Here, just the cells. So that's going to be our marker. That's our target. Here, with no embolin, so that's the untreated HSV1, treatment with 20 micromolars, comparable to the, yes, so this assay, I forgot to say, measures luminescence. Healthy cells can still metabolize and release ATP. So this assay measures the amount of ATP that's being produced by those cells. So 20 micromolars, comparable. 40 micromolars, still significant, but not as much. 54 micromolars, 60 micromolars. So and then showing we do statistical analyses as well. So there was a statistically significant difference in the luminescence or the amount of ATP that's being released when the cells were treated, when the virus and cells were treated with embolin. So attachment was inhibited. Then we looked at penetration. We also, again, the same thing, using luminescence as our indicator. It inhibited the penetration step. But post-penetration, after a longer period of time, once the virus got in, penetrated, embolin was not effective post-penetration. So we conclude for embolin to be effective, it's going to act on the early stages, put it in contact with the virus before it gets into the cell, and then it would inhibit replication. We also had a way of determining its antioxidant potential. And you hear a lot about products that are antioxidants. And antioxidants protect the cells from certain damage. And one compound that's released, a molecule that's released that's not good for cells is hydrogen peroxide. Hydrogen peroxide is released when the virus infects the cells. So antioxidants in this assay would reduce the concentration of hydrogen peroxide. And so Embolin protects the cells from reactive oxygen species, which can damage the cell. So peroxide again induced oxidative stress on the cells. So we treated both the cells and the virions with Embolin to determine the effect on lowering the amount of hydrogen peroxide and reducing the damage to the cells. We demonstrated with this assay that viral cells had lower hydrogen peroxide levels when infected with Embolin-treated HSV1 as compared to the untreated control. This is the virus only. And if you look at the bioluminescence indicator, again, the amount of hydrogen peroxide that's been measured, higher concentration, 25 micromolars. Here, comparable to the virus. But look, when we use higher concentrations, then we could reduce the percentage of hydrogen peroxide that's being released in the cells. So reducing the damage to the cells, comparing untreated virus with treated virus. Conclusions, once again, not cytotoxic, this plant extract Embolin, not cytotoxic at concentrations up to 70 micromolars. It reduced the infection of HSV1 in cultured cells. We could see that in the reduction in the number of viral particles released, and it reduced the oxidative damage that would have been caused by hydrogen peroxide. And the other area that we spent a lot of time on in my lab was plant extracts from green tea and from black tea. So again, plant extracts have been used for centuries to treat microbial infections. If you want to get some ideas about products to test, talk to people in other cultures in terms of what they did, what they used in the absence of vaccines and other antivirals. What did they do to try to keep people healthy? And so that gave you some ideas. So plant extracts have been used for centuries. It is believed that it's an evolutionary response to microbial attacks on plants. Plants have evolved ways of defending themselves from microbial attacks, and these polyphenols were used in that way. And then phenolic compounds contain numerous varieties of compounds, including simple flavonoids. So when we look at the tea extracts, tea is developed, the green tea, black tea, oolong tea, they're products of a plant, Camellia sinensis. And the amount of oxidation that the tea leaves undergo will determine which type of tea is produced. Green tea does not undergo oxidation. Black tea undergoes 100% oxidation. Oolong tea undergoes partial oxidation. Now, when we look at the green tea and black tea extracts, and we've done research in both of these in our lab, those polyphenolic compounds known as flavonoids make up a large composition of all three of those types of tea. But the major flavonol of green tea is the catechin, and that represents 80% of the polyphenols found in green tea. During the fermentation process, the catechins undergo a series of oxidative reactions, leading to the synthesis of the black tea extracts, which are theaflavins and thearubicins. The antioxidant properties of tea have been ascribed to the catechins. And green tea has been found to have more antioxidants than black tea. And when we look at the green tea extracts, epicatechin, epigallocatechin, epicatechin 3-gallate, epigallocatechin 3-gallate, or EGCG. The one that we worked with in the lab mostly was with EGCG. And EGCG was found to have antiviral properties against several viruses before we began working with. And it's always, this was brought to me by one of my coworkers, Dr. Lee, who was I would call her one of my collaborators, not just coworkers. We collaborated on this project, and one of her former students modified EGCG. Needed to modify it because EGCG, although it is antiviral, antioxidant, anti-tumorigenic, antibacterial, it's unstable, which means it's not going to stay around for a long time. It's unstable. So one of Dr. Lee's former students modified EGCG to make it more stable and lipid soluble. So I showed you the structure of EGCG, but then Dr. Stephen Hsu modified EGCG to EGCG stearate and made it much more stable, longer lasting, and lipid soluble. Black tea, theaflavins and thearubicins. So we have theaflavin 3-gallate, theaflavin 3'-digallate, and thearubicin. So the catechins undergo further oxidation to produce these black tea extracts. So we used both. We tested both against HSV1 and HSV2. Black tea was also found to have antioxidant, anti-cancer, antibacterial, anti-arthritic, and antiviral properties. But I was only interested in, of course, its antiviral properties. You've seen this setup before. We have to make sure it is not cytotoxic, so we do that in the 6-well plate. But we also wanted to test, at this time, compare acyclovir, green tea extracts, EGCG, palmitate EGCG, which is also lipid soluble, and EGCG stearate, and then black tea extracts. So I had a solution of combined black tea extracts with those various types of theaflavins. So we start out with the 6-well plate, treated the cells alone with acyclovir, with the extracts, the green tea extracts and the black tea extracts. And we wanted to determine, again, the maximum non-cytotoxic concentration that we could use. Then we added the virus. In the 96 well plates, We treated the virus with acyclovir. We treated the virus with the green tea extracts. We treated the virus with the black tea extracts. Then we used several assays to look at the effect. Cell proliferation is going to determine the number of cells that remain after treatment with either the compounds or the virus or the treated virus. Inverted microscopy, once again, we wanted to look at the cells so we can get a qualitative assessment of the effect of these reagents or compounds by looking at them with an inverted microscope. Then we did various antiviral assays, including the MTS, which is a the MTS, it's a colorimetric change. We look for change in the color in terms of when a product is being released. We did the ToxFlow again, which measures the ATP production. We also wanted to see, okay, virus gets in, what's the effect on the metabolism? So we could then extract viral DNA from the cells, and we could do PCR and qPCR to measure the products that are being produced after the virus has been infected the cells. And then as technology changes, we have to change as well. So we started using bioinformatic analyses as well, modeling analyses, get the sequence of the glycoprotein that's on the surface of the HSV, which a viral protein has to bind to that glycoprotein and start a reaction in which eventually the viral membrane will fuse with the host cell membrane, and then the virus will enter the cell. It's called endocytosis, but the viral membranes have to fuse. So that first step is binding to glycoprotein D. So we did bioinformatic analysis to model or get an idea about how efficiently EGCG stearate, and that's the one we used most often because that's the most stable, will bind to viral glycoprotein D. And our results, both EGCG and the lipophilic EGCG, the modified forms, not cytotoxic to Vero cells, which are the monkey kidney cells, and the A549 cells, which are the human fibroblast cells, at concentrations up to 100 micromolar. The black tea extracts that consisted of greater than or equal to 80% theaflavins, not cytotoxic to Vero and A549 cells, at concentrations up to 100 micromolars. No effect on the cell morphology. And again, we looked at that with the light microscope. We just wanted to look at the cells and make sure there was no change. Take an image of the cell. No effect on cell viability. It was not cytotoxic to the cell, and it did not prevent the cells from continuing to divide. So the conclusion, tea extracts are safe. Yes. All of that yes, tea extracts are safe. So with cell viability, you can see that. You've seen my graphs before. Percent viability, look at the cells alone, EGCG, EGCG stearate, DMSO, and in this assay, we looked at ethanol, and these are the various concentrations. Cell viability, again, safe. Then with black tea extracts, the same. In A549 cells, 1.4 millimolar concentrations, 0.014, whatever concentrations we used, black tea extracts were also safe. Then we were concerned about the percentage of inhibition. Did it work? Did it work? So we measured that with luminescence. So we treated the virus with different concentrations of EGCG. 75 micromolars gave us the best results in terms of the percent of inhibition. On the y-axis, percent of inhibition, increasing concentrations of EGCG steroid. With the black tea extracts, I had another student who did a project with the black tea extracts and looked at black tea extracts in synergy with acyclovir. Let's use both. Put both acyclovir, BTE on the cells. What works best? Again, they work by different modes of action. The black tea extracts targeted attachment and penetration. Acyclovir, the enzyme, the replication enzyme. Let's combine them both, see what happens. And so when we looked at the combination, again, most effective when TF3 alone, theaflavin 3, or the black tea extracts, or the black tea extracts in combination with acyclovir, the greatest percentage of inhibition when they were combined, when they acted in synergy. Again, to bring virus infections under control, you have to attack them by multiple means. Multiple means if you're looking at antivirals. So the plaque-forming units, remember, that's the titer, that's reducing the count. And so when we looked at the number of viral particles, untreated, you see, this is our untreated control, the number of viral particles when we did not treat the virus, but look at the reduction when EGCG alone, 50 micromolars of EGCG was used, we reduced the number of plaque-forming units, but 50 micromolars of the palmitate EGCG, which was the lipophilic version, 0 in terms of the plaque-forming units. And I mentioned before, another thing that we did, and this was earlier in our studies, I had the students learn to extract the viral DNA, which means once you know the sequence, you can make what are called primers. Primers will start the process of copying or bringing out the DNA that has those sequences. So we have primers that go in either direction. So, and here, that's just the ladder to give us an idea about the size. And PCR products that were extracted from HSV1, A549 cells treated with 1.4 millimolar in columns 2 through 4. When you compare that to untreated in columns 5 through 7. So we had 2 different glycoproteins. We had the sequence for glycoprotein B and glycoprotein D. But you can tell when you treat the virus with the black tea extract, no viral DNA here, smaller. So when we look at the intensity of the bands, it's less intense than the untreated glycoproteins. And then in the bottom, a different student did this one in panel B. Column 2 is the untreated, and then we have increasing concentrations of black tea extract. So it reduced the intensity, did not eliminate it completely, but reduced the amount of viral DNA that's released. And then real-time PCR analysis on HSV2. The same thing. We looked at extracting the DNA. Here is the untreated HSV2 control. This is when the HSV2 was treated with EGCG steroid. And then, of course, you would not expect to find any viral DNA in just the viral cells alone. So it reduced the copy number or the number of molecules being produced in the PCR reaction. And again, my favorite with the fluorescent or confocal microscopy. Blue, again, to stain just the nucleus. The green represents, and if you look at the shape of the cells, you see when the virus infects here, the shape of the cells, the morphology of the cells changes, but then we see, and this is only after we would extract the DNA 12 hours post-infection, because we didn't want it to lyse completely and we lose it. So we would extract the DNA 12 hours post-infection. So just 12 hours post-infection, you see the expression of green fluorescent protein. We had the green fluorescent protein in HSV1 and in HSV2. So in both of those, we could look for the expression, and then once we treated the virus with EGCG steroid, then the expression of the green fluorescent protein is reduced. And then the last thing that we began looking at is using bioinformatics. Bioinformatics is modeling. And so once you get the sequence of the glycoprotein D, then you get the structure of the EGCG steroid, you wanted to see how it fits into the receptor. On the surface of cells, and I call it the door or the pathway in which the virus can enter. And so you wanted to look at what's at that door. What's at that door? And can this molecule somehow block that door? And so the modeling will tell us whether or not, or how likely it is that it could. And so we did bioinformatic analysis to look at how the EGCG interacts with the viral proteins. So it fits right in there. So the bioinformatic analysis, and this is just telling what type of bonds are formed between the molecules and the compound that we're using and the protein, the viral proteins. So bioinformatic analysis showed us that the EGCG steroid can interact with glycoprotein D. Again, EV69, and that's the last one, not the last one, but this is the one that I said we had more difficulty with. EV69 is naked, which means doesn't have a membrane on the surface, but on the outside, it's just that nice protein shell. The protein shell serves a function, that is to deliver the genetic material to the next susceptible host. And so those naked viruses that don't have a membrane, it's a little bit more difficult. They're much more resistant. So we tried EGCG steroid on EV69. Did not impact the ability of it to bind. It did not. So we continued to look. Well, it had some effect because it reduced the number of viral particles. So what was that effect? It was the antioxidant effect. So we did the Ross-Glow assay again, where we're looking at the reduction in hydrogen peroxide. Hydrogen peroxide is going to be damaging to the cells. So we're looking at whether or not EGCG steroid could give this protective function to these MRC5 cells. They're also lung cells. Again, so the EV69 can replicate in those MRC5 cells. And then we found again, when you compare the untreated EV69 with the uninfected cells or with the various concentrations, we could reduce the percentage of, or the hydrogen peroxide levels at specific concentrations. 25 micromolar, 50 micromolar, 75 micromolar. Untreated EV69 had a higher level of hydrogen peroxide. So the results of our tea experiments using black tea extracts and the green tea extracts, EGCG, lipophilic, which is the lipid-soluble EGCG, they were both effective in reducing the infectivity of both HSV-1 and HSV-2. The tea extracts blocked the production of both HSV-1 and HSV-2, and that's what you want to do. You want to stop it from releasing more viral particles that will then float around in the air and on the surfaces and come in contact with another susceptible host. The tea extracts will block the attachment and absorption or the penetration of HSV virions, and virions are the viral particles. It will block the ability of it to attach, block the ability of it to enter the cells, both viral cells and A549 cells. EGCGS, which we test for its antioxidant abilities, provided protection from EV69, which is the naked RNA virus, EV69 mediated oxidative stress. I mentioned that I had a graduate student to look at black garlic, and I did not bring that data, but and we just had a paper published with the black garlic extract. And so the black garlic, again, was the fermented form of the garlic. It is better than the unfermented form because it contains an increased level of antioxidants as compared to raw garlic. And my student determined that 13% of the black garlic extract, which was suspended in ethanol, alleviated oxidative stress. And the hispalons. We just submitted a paper with the hispalons. I was contacted by 2 chemists in India, and they said, well, we have these compounds. We have 6 classes of compounds that we wanted you to look at. I said, okay, no, 4 classes of compounds that we wanted you to look at in terms of the antiviral properties. I said, sure, send the 4 classes of compounds. And what arrived was 24 different compounds, 4 classes, but 6 different variations. I said, oh, we've got our work cut out for us on this one. And we took those 24 compounds, we tested all 24 of them, then different analogs, different structures. Out of the 24, I found 1. One that was antiviral. And so we did our same assays, 6 well plates, 96 well plates, antiviral assays, antioxidant assays. And so we just submitted a paper showing the results of that. And with that, our conclusions, overall conclusions. Plant extracts have the potential to act as antiviral agents. They have the potential because, again, the antiviral agents must be effective without hurting the cells. So we found them to be non-toxic, and especially they could be applied topically. Some are looking at their laboratories that are looking at using these in topical applications. And they can, when you apply topically, can reduce or block the production of infectious virions. The mode of action inhibits the early stages. The plant extracts from green tea, black tea, curcumin, embolin, safe, and could be developed as their potential therapeutics. Oh, okay. Work was funded by the MSU Faculty Scholarship Program, the Lustigman Scholarship for the student research. And again, most of this work was by students. Lustigman Scholarship, the Science Honors Innovation Program at Montclair State. And then we thank Dr. Andrea Bertkey, who provided the HSV to GFP. All of the student researchers involved are listed. My collaborators, Dr. Lee from Montclair State, Dr. Chu from Seton Hall University, Dr. Su from Dental College of Georgia, Miriam Rossi, Franco Caruso from Vassar, Kevin Billick, I'm working with at Montclair State now, and Dr. Shubaraju and Ramani from Andhra University in India. And with that, thank you.
Thank you so much, Professor Adams, for such a wonderful and very thought-provoking lecture. We have microphones that we are passing around. So if you raise your hand as you're starting to, we'll get microphones to you. And just wait till you get a microphone. And then to those online, if you raise your hand, we'll try to get to some of those people online as well. And why don't we go? I see someone right up. See her up there in the back?
Yes.
Yeah. Hold on for a second. Your mic might not be on.
Thank you for your talk this morning. It's been enlightening.
Can you speak a little louder for me, please?
Thank you for your talk this morning. It's been enlightening.
Thank
A whole foods plant-based diet sounds like a good idea to me personally, based on a few documentaries I have seen, like Forks Over Knives and the book How Not to Die by Dr. Michael Greger. I've tried to read a book by Dr. Colin Campbell called The China Study, but it was a little over my head scientifically. So I was just wondering if you had any thoughts on a whole foods plant-based diet.
On the plant-based?
A whole foods plant-based diet.
In terms of the plant-based diet, plant-based diet, again, I would just say in terms of the antioxidant potentials, that would be good, as long as you make sure you have all of the other nutrients as well, that, the other proteins that are required and the other vitamins, but the antioxidant potentials of plants are extremely helpful.
There's a question right in the middle, right there.
Yes.
Hey, thank you for the presentations, much appreciated. So the question I have, the compounds you were using, the extract, a couple of questions. Did they test it against organic and all the materials like the turmeric that you were using? There's a difference between organic and GMO modified for the plant extracts. Also, I see you using DSMO, but if you look for DSMO, it's not recommended. A lot of places don't want you to use DSMO. They say it's toxic to the body. So if you go out there and try to use turmeric, people may use it, but turmeric oxidizes very quickly.
Yes.
So you can use, even if you use an organic materials, if you don't use it quickly, it oxidizes. So if you think people are going to go out and use turmeric, there's all different forms of it. And I didn't see you have a control study of organic based or GMO based.
Yes.
And also mentioned about why, I guess that's the first part of the question. The other part of the question is when all this stuff was going on and people were getting sick and they were using vaccines, you didn't hear anybody saying using natural, and you're getting very good results using plant extracts, but you don't see that. And you don't see that as a substance put into the vaccines. So you would think that if science was moving forward and all the research that you did, that they would start using that more readily. So I just want to know what your comments were.
When you look at antivirals and vaccines, again, act completely differently. And so when you look at how the vaccines have been developed through the years and compared those to that's why I started out with the difference between the vaccines and the antivirals. But when the vaccines initially were started out by using either the weakened live virus or heat-killed virus. We've progressed beyond that to taking just a part of it. The goal again is to induce that immune response. My preference again, start out with vaccines. Start out with vaccines. I'm a proponent of vaccines. I know there is a lot of anti-vax talk out in the general media, but when you look at the vaccines, and even though they may not recommend, like we use DMSO in the lab, we also use ethanol, so they can be suspended in these polar solvents, DMSO as well as ethanol. But there have been studies, again, in terms of the percentage that can be safely included in compounds without any negative effects. I don't know if I answered your question, but
Just the question on organic versus GMO modified.
organic versus GMO modified. Again, I don't have a problem with GMO modified, so it's, yeah, I honestly don't. But again, that's not my area. That's my personal feeling.
So there's a student with a microphone to your left, right,
Yes.
right there. See him?
I was just gonna ask, when you said that viruses can travel on surfaces and through the air, I learned that viruses travel through aerosols. So how would they be traveling through the air if they're in aerosols? I just was
Well,
wondering that.
when you say aerosol, so what's in air? So what's propelling them? So there's something called, they collide. So the more viral particles that you have that are released, the greater the distance that they will travel. And suppose they just settle on a surface, and that's what will happen. They'll settle on a surface. They'll settle on a surface. You touch it with your hand. Where does the virus go?
Your hand
On your hand. Where does your hand go?
and your face.
And so, yes, and then what happens? You sneeze, you propel them, you cough, you propel them. I speak loudly. You speak loudly, you propel them. You sing, you propel them. So they are flying through the air, waiting for a susceptible host. And once they find that susceptible host, and if it is a virus that can replicate in that cell, it'll set up a productive infection unless it has been seen previously by the immune system.
So in that case, if they travel and land on a surface, how long will it take for them? How long do they have to get into a host before they
It
die?
varies. It depends on the virus. The enveloped viruses, with that membrane, the membrane may be more susceptible to, that may not survive as long. But those naked viruses, when you, like on I call it the cruise ship viruses that are on every surface, those are naked viruses, protein only, and the protein's function is to protect the genetic material until it can get to another host. Okay.
There's a question right in the aisle, right there.
Yes.
The great,
Oh,
it was,
good.
there we go.
I
It was a
need that.
great talk. Thank you so much. Have you or are you aware of other people studying compounds in coffee for the same properties?
I will be honest, you know, as I said, we started out, I had a student, I did a project with a high school student initially. We looked at caffeine and we looked at the caffeine. And so when you think about coffee, but I don't know of any other studies in which they looked at any other compounds extracted from the coffee. But we did, I had a high school student to look at caffeine in a virus that was very benign and that he could work with. And the caffeine inhibited that virus from replicating.
So right here in the front.
Thank you very much. That
Yes.
was absolutely fascinating. So if you were taking antivirals while being vaccinated, is there possibility that that will reduce the the effectiveness of the vaccination?
Oh, it would not reduce the effectiveness of the vaccine. I would not say that the antivirals would reduce the effectiveness of the vaccine. The vaccine, again, the goal is your immune system will see it. will see that pathogen again and then immediately produce antibodies and T cells to target the vaccine. But during that time, when you're waiting for it to build up that immunity, you're shedding virus. And if you're shedding virus, then you want to try to stop that so that it does not get to another host.
But if the antivirals are stopping the shedding of the virus and entering the cells, does that not reduce the?
Oh, so the question is if the antiviral is binding to the virus and blocking it from entering the cells.
Will that stop the immune system recognizing that virus?
But the immune system is already, well, if that was the very first one to get in, then it might not yet, but it's
Yeah,
not,
okay.
that was not the very first one to get in. It said the virus has already gotten in. The immune system has already started trying to battle it. And in doing that, again, some of them still might escape until you build up that full immunity, until you have enough antibodies and T cells to bring them under control.
I see. And just since I have the microphone,
That's
just
fine.
I should keep drinking afternoon tea.
You should keep drinking
Afternoon tea.
hot tea,
Hot tea.
hot tea,
Hot tea.
hot tea. And the reason that
That's
I
what we
say
do.
hot tea, because again, those compounds, it's was mentioned it, them not being very stable. And so they're not going to stay around. So when you buy these bottled teas, they taste good. And, but the catechins are gone. So the flavor is there, but the catechins no longer exist. Brew your own.
Yep.
Brew
in a
your
teapot.
tea.
In a
Yes,
teapot.
brew your own.
Hence the rise of the British Empire was all done by tea.
That-
There's a there's a question in the middle.
I have a question about tea also.
Mhm.
You mentioned the different teas. You have the black, the oolong, the green tea, and one has the antioxidants, and one has no antioxidants.
Oh, no, they all have antioxidants. They differ in the amount of antioxidants.
Antioxidants. Well,
Yes.
that's my question. You hear everything about antioxidants, and that's something I'm unfamiliar with. So could you address the green, black, and the antioxidant? Is it good for you, bad for you? How does it operate?
Oh, I think the antioxidants are good for you. The antioxidants in teas are good for you. Again, the antioxidants, whether it be the green tea or the black tea or the oolong tea, I wrote that hasn't come out yet. I mentioned giving a talk at UC Davis I may not have mentioned that, last January, and it was a tea institute, the Global Tea Institute at UC. They studied the Global Tea Institute at UC Davis. So I submitted a chapter on the health benefits of tea. So they're trying to put out a book on that one. But no, continue to drink the tea, whether it be green tea, oolong tea, black tea, continue to brew your tea and drink the tea. The antioxidant abilities, keep saying neuro, the cytoprotective effects make it well worth it, as well as from your plant-based diet, those antioxidants are good for you.
We've got two questions. We've got time for a few more. We got two questions in this side. There's one here and then there'll be one over here. And I know we're going to end over there. So right there.
Okay, thank you for your talk.
Mhm.
So does the antiviral process ever eliminate the virus entirely from the cell, or does it just suppress it at a low enough level so that it doesn't create illness or disrupt the
Well, a
remaining cells?
lot depends on the viral load as well. Even acyclovir, which has been approved, FDA approved, acyclovir is very good at stopping that virus from replicating. But again, because I work with herpes viruses, you cannot eliminate herpes viruses once you're infected. You cannot eliminate those. So the antivirals are good at targeting the virus, stopping them from getting in, stopping certain processes from replicating. I can't say that every antiviral or any antiviral would stop all viruses from replicating. There's not a one size fits all. So, and then you'd have to determine what's the effective dose. So that goes beyond what I do. I simply look, does it have any potential? Is it worth investigating further? And we either a yes or a no on that. And so the ones that I've reported on, I give a yes on those.
And then to your right, about halfway up, right there.
Thank you for your talk.
Yes.
Any follow-ups with animal studies?
Oh, I don't do animal studies.
Anyone?
The Hispalons that I was working with, the one that that we just got a paper on. Now he is planning to move into animal studies. I believe there was a clinical trial in, not going to say for sure, in China using the stearate in terms of a topical. That one I'm not quite sure about. But I do know that would be, of course, one of the next steps. Once you identify in cell culture, then you find an animal model that it could possibly work on.
Thank you.
Okay.
Anthony, we have one question on Zoom. Ashok, if you are still there, can we unmute Ashok?
Yep. Hi, Dr. Adams. Excellent talk. Really enjoyed it. Ayurvedic
I don't hear
treatments in
anything.
India. Hello, can you hear me?
Going once,
Can you hear me?
going
Hello?
twice.
Oh,
Sorry.
So our last question will be right here in the middle.
Yes.
Thank you very much for the presentation.
Thank you.
First, I'd like to appreciate you for your excellent work and also for your students. Thank them. And I also wanted to commend you for acknowledging that if anyone wants to do a study, check on what indigenous people do.
Yes.
And I want to say that because I am a product of my indigenous upbringing for plant medicines.
Yes.
So for me, my first go-to will plant medicine.
Mhm.
The vaccine will be my bottom.
Yes.
Okay. Got it. So, and other questions, I had the rest of the people who asked questions already, and so it's answered. And at least I know where you stand in that. My question would be, we've been discussing about the physical properties, physical effects on our bodies. Although there's a good suggestion for animal studies, and I believe there are studies out there.
Yes.
And you may not be doing the body physical with actual human studies, but would you be willing to for other collaborations with those who will do it? Not that I needed a convincing, because I'm fully convinced already, but for those who need intellectual masturbation, this is a perfect study.
Yes.
So my primary question is, would you consider also expanding your study in terms of quantum morphogenetic physics or science or biofield? Because in quantum science, we are more than physical body. Your study is very controlled, but we live in a life that is not controlled. And for my friend here, we have to remember also that creation has that balance.
Yes.
Antioxidant is overemphasized. There's a need for our body to have that oxidative process,
Yes.
but there's a need for the antioxidative. So I just want to challenge your team to please work with other people who may not be on your same perspective.
Yes.
Because that will help us really be together. And of course, I would like to invite you to my podcast, Quantum Nurse Podcast, because I feature world-renowned scientists. When you mention about vaccine, there is Professor Denis Rancourt from Montreal, and he has this all-cause mortality that proves that vaccine was not effective. But I want to provide a place for you because people believe on plant medicines. So I will give you my card.
Okay.
Thank you.
And so with that, I would also say in terms of, I study one section. Someone else, as you said, animal studies. In order to get anything to the market in terms of it, you have to start out with controlled studies. Start out in the lab like I do. Then someone else takes it from there, moves it into animal models. Then from there into, develop it, but dosage, a lot of, my lab alone, we're not equipped to do that. As I said, I was not equipped. My role is to teach, and my role was to teach, and that's what I did. And so you take that information, you release that information in terms of publications. Someone else will pick that up. Maybe this is something that I should do more with. And then I would be happy to answer any other questions about that. But again, it's not isolated, as you pointed out. It is not isolated. You have to look at the entire picture and then put all of the evidence together and then come up with that conclusion. Okay.
Thank you so much, Professor Adams. Next week, we go on to climate change and the path to net zero energy. We'll see you next week. Thanks, everybody.